We're recording it to the local. Yes.
Okay.
Okay, folks. Sorry.
Sorry. No, go ahead.
Welcome, everyone. I'm here today with Dr. Chuck Huber from Stata. And thank you very much, Chuck, for joining us this afternoon in Toronto. I'm sure it's morning time where you are.
Yes.
And welcome to everyone. We have 135, 38 people registered. We already see about 35, 40 people in present, and it's already a few minutes over, 12:04, so we start in a minute. We are quite pleased with the fact that Stata has been kind to support this these learning initiatives that we have from Toronto Metropolitan University. A bit of introductions. My name is Murtaza Haider, and I am a professor at the Ted Rogers School of Management at Toronto Metropolitan University. We used to be known as Ryerson University. In the last year, university changed its name. So now we don't have to tell you that we are in Toronto because we are Toronto Metropolitan University. So that's a good advantage of changing the name with the city in it. We are the largest business school in Canada, the Ted Rogers School of Management. We have 12,000 full-time equivalent undergraduate students that make us perhaps larger than many universities in Canada and North America. And our focus is on all aspects of research in management and business. And one of the key things that we do is to promote literacy in empirical methods, research methods, given that data is front and center to whatever we do in business and economics and management. So we are always working with data and platforms to analyze data a little better. And one of the things on the research side is this benefit of or this technique called meta-analysis, where one can use existing research and existing parameterization of others' work that people have done research, ran models, and they have reported parameters in their publications. Can we use that as our data to resolve questions, to find answers to questions? So there are many questions for which research gives you different answers. One research says that increasing the interest rates would have such an impact. The other one would say, no, increasing the interest rates may have another impact. So a lot of discussions on those. And when you have this conflicting conclusions in research, meta-analysis seems to be a useful tool to figure out where the weight of these arguments reside. As I said, we are fortunate to have Dr. Chuck Huber with us from Stata. Stata incorporated Meta a few years ago. It's a very powerful tool now available in Stata 17. I think it was a few versions earlier. So all of this will be discussed today, and I'm going to pass it over to Chuck, and he will be sharing his screen with slides. And so I want to make sure that he can share his slides.
Yes.
Let's just do that before I pass it over. So there you go. I think he can. So
okay,
thank you, Chuck. Over to you, sir.
Okay, great. Thank you so much for that nice introduction. I'm going to see if I can stop my video so that it's easier on the system. So yeah, my name is Chuck Huber and I work for Stata Corp. And today we're going to talk about meta-analysis. A little disclaimer, I just rewrote this entire talk in January, and you're only the second group to see it. And so if I seem a little lost in my own slides today, it's because I'm so used to the old version of this that I would probably get lost in my new slides. But I like the way this one flows better. I also, it seems, need to update my title slide because I still have Ryerson University on here and I need to update that. So I'll get that changed for the next time I do this.
Thanks, Chuck. May I also say that for those who are joining us now, if you have a question, it's a webinar, you won't be able to unmute yourself, but you can put your questions in the chat. And I will be monitoring chat for Chuck, and any questions you may have, I will then pick them up from there and then pose it ask of Chuck for you. So please put your questions and comments in the chat box, and we will be answering throughout the presentation. Thank you, Chuck.
Yes, thank you. And I'll be happy to hang around after for a while and answer questions as well. So if you'd like to download the materials, you can grab the slides, the data sets, and the do files here at tinyurl.com/Stata Meta-Analysis. So all these materials are located there and you can grab them. So here's what we're going to talk about. We have a lot to talk about with meta-analysis, and I'm not going to read the whole outline because it's so much. I'm just going to dive right in, but we have a lot to cover in a short amount of time. I do want to start off with an acknowledgement slide, though, because we have had meta-analysis in Stata for a long time, but the original suite of commands was actually written by our users. And I'm not going to read all their names here, but I've listed them on the slides. These are people at different universities around the world who wrote the original commands that we based our commands on. All of the modern official Stata commands were written in-house from scratch, but they were inspired by the work of a lot of people around the world. And that's part of the reason we took so long to include meta-analysis in official Stata, because the user written commands were so complete and so good that there really wasn't a big reason or a need for us to include it in official Stata. But we did a couple of releases back. And so what I'm going to show you today are the official Stata meta-analysis commands. Okay. So let's start with a question. I like to make my presentations very practical. So I'm going to start off with a question, and that is, does teacher expectancy have an effect on pupil IQ? In other words, do the expectations or the beliefs of the teachers, their beliefs and expectations about the students, actually have an effect on their IQ tests, the students' actual IQ tests? So that's the question. And this question actually started back in 1968. There were a group of researchers, Kahn, Edwards, Rosenthal, and Crown, asked this question, and they published a study in the journal Psychological Reports in 1968. And they found that, I'm paraphrasing, that essentially, yes, there is some effect of the teacher's expectations or beliefs about the student's ability on their performance on IQ tests. So 1968, this group found that there was a relationship here. 1969, William Claiborne from Syracuse University tried to replicate this study, and it failed to replicate. So that was interesting. It's always strange when you have one study that reports one result and you have another study that fails to replicate that result. So 68, then 69 doesn't replicate. 1971, another group tries to replicate this study and has sort of mixed results. So 1971, there's a replication. 1972, there's another study that has somewhat different results. And by 1984, there had been multiple studies done of this, trying to answer this question with sort of mixed results. And so Stephen Raudenbush from Harvard University did what we now call a meta-analysis. And he was looking at what are the study level factors from these various experiments and studies that seem to influence the outcomes of these different studies. And so one of the things, this is a table from Raudenbush's paper, where he lists the various studies and their results, but he also lists various attributes of the studies. So, for example, the studies didn't all use the same IQ test. Well, that could certainly affect the results. If you don't use the same IQ test, you might get different results, not because of the relationship between the teacher's expectations and the students. Maybe the differences are due to using different IQ tests. Or maybe some of these were blinded to the test administrator and some of them weren't. Some of them were group testing versus individual testing. And so there may be different differences in the way the studies were conducted that affect the results. And so that's one of the questions that we can ask with meta-analysis, is going sort of beyond. First off, we'd like to answer the question, what is the answer to the original question? But we also might want to explore what are the attributes of the various studies that have some sort of an effect on those results. And so that's one of the things that we can do with meta-analysis, is exploring this heterogeneity of the results across the studies and what are the things that may be affecting that. That study also used a variety of what we would now consider fairly crude statistical methods, basic chi-square tests and things like that. We have far more sophisticated techniques now for doing formal meta-analysis. So these were some of the early studies that might be called meta-analysis. So where does this term even come from? Well, this is Gene Glass. He is the person who coined this term, meta-analysis, in 1976 in a paper called Primary, Secondary, and Meta-Analysis of Research. So he's the person that coined this term and sort of formalized this. And he suggested that we could use these kinds of techniques to provide unified answers to a variety of research questions. Things like, does taking vitamin C prevent colds? Does exercise prolong your life? Does lack of sleep increase the risk of cancer? Does daylight savings time save energy? And all kinds of questions like this. And it's actually grown to the point where there's actually a large repository now called the Cochrane Project. And there's a website you can go to that is a repository of all of these meta-analyses, and many of them will allow you to download the meta-analysis data and explore the raw data on your own. Not the raw data from the original studies, but the meta-analysis data. Okay. So one obvious question is, does it even make sense to combine the results from different studies? And Borenstein is one of the original people who worked in this area, and he has a famous quote from this book. It says, in the early days, Robert Rosenthal is the person the quote is from. In the early days of meta-analysis, Robert Rosenthal was asked whether it makes sense to perform a meta-analysis, given that the studies differ in various ways and that the analysis amounts to combining apples and oranges. And Rosenthal had a clever response. He said that combining apples and oranges makes sense if your goal is to produce a fruit salad. And so that's one of the questions we really need to ask ourselves as we're doing our meta-analysis. Are we really trying too hard to combine apples and oranges and we're simply making a fruit salad? Or are we distilling the results of a bunch of apples into some really good apple juice, I suppose, is another way of looking at it. So we have to keep in mind that it may not be, one of the things we have to pay special attention to is it may not even make sense to combine the results from multiple studies. So that's one of the questions we'll be answering along the way. Okay, so these are the goals of meta-analysis. First is if it's sensible to provide an overall estimate of the effect. What is the answer to the original question these studies were trying to answer? And then secondarily, to explore the between-study heterogeneity. Studies often report different and sometimes conflicting results, both in terms of the magnitudes and the direction of the effect. And if that's the case, why are they different? It might be if the studies are too different, the question then becomes, why are they different and what are the factors that lead to those differences? And then the third thing that we're going to focus on is evaluating the presence of publication bias. And we'll talk more about that later and define it, but it's essentially the underreporting of non-significant results in the literature. If your p-values are greater than 0.05, you're not likely to publish them. And so that leads to this bias in the literature where we only see studies that have p-values with p-values less than 0.05. Okay.
Chuck, can I ask you to show the slide with the slides information where people can download the slides that's been requested?
Oh, absolutely. Right here.
So it's tinyurl.com Stata Meta-Analysis.
Yes.
Thank you.
Yes, I probably should have pasted that into the comments window earlier. That might have
I'm doing it right now. So
okay, great, thanks. Okay, let's see, where did we leave off? Okay, so there are a variety of components of meta-analysis, and so these include the effect size. What is the effect size? Is it a difference in means? Is it an odds ratio or a risk ratio? There are different models we can use for doing our meta-analysis. We'll take a look at those shortly. There are different ways we can summarize the data, whether it's in tables or something called a forest plot. We also need to look at the heterogeneity or the differences between the effect sizes across the studies. We need to look at small study effects or systematic differences between the effect sizes reported by small versus large studies. And then we're also going to explore publication bias, which I mentioned a moment ago. So what I thought I would lead off with is just a quick summary of the commands in Stata that are available for these different purposes. And we're going to go through each of them. But just as a quick summary, for declaring your meta-analysis data, there are a collection of commands: meta-set, meta-esize, meta-update, and meta-query. Those have different purposes, and I've listed the descriptions here. And again, I'm going to go through them in detail shortly. For summarizing your meta-analysis results, there's meta-summarize and meta-forest plot. So you can create either tables or graphs of your meta-analysis. And then for exploring heterogeneity, there's meta-summarize by group. So there's a subgroup option. You can use the subgroup option also with meta-forest plot. There's also meta-regression. You can generate predictions of random effects. There's EStat bubble plot and Meta-Lebesgue plot. I think I'm pronouncing that correctly. And so these are some of the commands that are available for exploring heterogeneity among the studies. For looking at small study effects and publication bias, there's the meta funnel plot command, meta bias, and meta trim fill. And then there's also cumulative analysis, meta summarize with the cumulative option and meta forest plot with the cumulative option. So those are the commands that we're going to be focusing on today and kind of how they fit into the overall framework of meta-analysis. There's actually a large control panel that walks you through this process. So if you go under the statistics menu in Stata, if you scroll way down the list where it says meta-analysis, if you click that, it will open this meta-analysis control panel. Let me zoom in on it so it's easier to read. And this will essentially walk you through the process of doing your meta-analysis. So if you start at the top and click on the setup button, that will populate the control panel with the features that are available for setting up your meta-analysis data. And then we can click on the summary button and the forest plot button and other buttons, and that will populate the control panel with the appropriate features. And so we're going to walk through this and actually do a meta-analysis. So preparing the data, the first thing we need to do is declare the data as meta-analysis data in Stata. This is a bit like doing panel data analysis or survival data. We're going to actually declare the features of the data so that we don't have to keep telling Stata about those features over and over again in each of our commands. We're going to declare the data or meta set the data first. And once we've done that, then it makes the other commands much easier to use. So we're going to use meta set and meta esize meta set will simply declare pre-computed effect sizes, and meta esize will actually compute and declare the effect sizes from summary data. So if I'm in the control panel, if I click the setup button here at the very top, it says declare meta-analysis data. So one of the choices here is to compute and declare effect sizes for two group comparisons for continuous outcomes. So let's say we have a continuous outcome of some sort. We could select that, and then we can select from a group of possible effect sizes that we've already computed. Things like Hedges G or Cohen's D or Glass's delta 1 and 2, or just raw mean differences. So there are a variety of effect sizes that you could choose from and that you've already computed, and you could simply put them in. So in doing this, you're going to have to tell Stata about the sample size, the mean, and the standard deviation for each of the groups, usually considered the treatment group and the control group, or you could just call them group 1 and group 2. So that's how we would set this up using the control panel, or you can use the command at the bottom where it says meta esize. N1 is the sample size in the first group, M1 is the mean in the first group, and SD1 is the standard deviation in the first group. And then N2, M2, and N2, M2, and SD2 are the same corresponding to group 2. And then we specify the effect size. In this case, I've selected Hedges G as the effect size that we've already computed, and then we would have our data set and be able to go on about our meta-analysis. Just to emphasize, there are two broad categories of these effect sizes. There are the unstandardized effect sizes, where it's just delta, or the difference between the means is just the raw difference between the mean in group 1 and the mean in group 2, or the mean in the treated group and the mean in the control group. But there are the others, such as Hedges G, Glass's delta. Those are considered standardized effect sizes. So that delta is the difference between the two group means divided by the standard deviation. So just so you're aware, Hedges G, Glass's delta, some of these have different names. If you're familiar with meta-analysis, you may be, or effect sizes in general, you may be wondering, why is there a Glass's delta 1 and Glass's delta 2? Well, Glass's delta, remember Glass is the person who developed these methods and coined the term. His original definition of Glass's delta was it's the difference between the mean in the treated group and the mean in the control group divided by the standard deviation in the control group. But that's only defined for experimental designs. Sometimes we have observational studies and there is no control group. We just have two different groups. And so in Stata, what we've done is we've defined Glass's delta 1 and Glass's delta 2. And the difference between the two is simply what the standard deviation that we divide by in the denominator. So glasses delta 1 divides by the standard deviation in group 1. Glasses delta 2 divides by the standard deviation in group 2. So that's the only difference between them. They're standardized by different standard deviations. The other choice here is to compute and declare effect sizes for two group comparisons of binary outcomes. So if we have a 2x2 table, again, we would specify, in this case, the number of successes and the number of failures in the treatment group, and then the number of successes and the number of failures in the control group. So these could include things such as, these effect sizes could include things like the log odds ratio, the log risk ratio, the risk difference, or a log Peto's odds ratio if you prefer to use that. Again, there's a command for doing this. If you use the control panel and hit go or OK, what it will do is issue the command meta e size, and then you just tell it the number of the count in each of the cells of the 2x2 table, and then select the odds ratio that you've already pre-computed. Okay. Then the third option is to declare generic pre-computed effect sizes. So this is if it's something you've done that's a little off the grid, you can just select whatever your effect size is that you've computed, and you can either specify the standard error or you can specify the confidence interval. So if you have your own effect size of some sort, you just enter whatever that effect size is, and either the standard error or the confidence interval. And again, I've put the command syntax here at the bottom just so you can see how that works. So there are a variety of ways you can tell Stata about the structure of your meta-analysis data. The other thing that you're going to need to specify is a model for your meta-analysis. What sort of a model do you want to use? The default is to use random effects, and we'll come back to why that is later, but this is the default, and it's a very commonly used model for modeling meta-analysis data. And then once you select the model, there are a variety of methods that you can use. Restricted maximum likelihood is the default, and again, it's probably the most commonly used. But there are other methods, maximum likelihood, empirical Bayes, and some others. I'm not going to list them all off. They're used in certain circumstances, whether you have very small sample sizes, if you have a small number of studies. Some of them are biased in certain cases and unbiased in other cases. And so usually maximum likelihood with using restricted maximum likelihood works pretty well in most cases, but there are other options included in case you have a use for those. Okay, so let's take a look at our data. We have this actual data from the Raudenbush study, that 1984 study that evaluated the effects of teacher expectancy on pupil IQ. So this original study in 1968 discovered what they call this Pygmalion effect, the idea that the expectations of the teachers had an effect on the outcomes of their students. And later studies had trouble replicating this. And so in 1984, Raudenbush published this meta-analysis that I showed you earlier, and we have the data here. So the data is called Pupil IQ, and you can download this from the Stata servers by typing webuse pupil IQ. And this data set contains the following variables. We have study LBL, which is the study label. We have STD-MDiff, that's the standardized difference in the means. So it's the difference in the means divided by the standard deviation. And then I have the SE, the variable SE is the standard error of the standardized mean difference. And then I have a couple of other variables. One is named weeks. Weeks is the weeks of prior teacher-student contact. How much contact did the teachers and students have? How many weeks did they have prior to the IQ test? And then week 1 is a categorical variable, and it's an indicator for whether or not the prior teacher-student contact was more than 1 week. So we have student-teacher contact as a continuous variable measured in weeks and as a binary indicator for whether or not the contact was more than 1 week. So that's the data that we're working with. So this is just a listing of the raw data. So again, study LBL is a listing of the actual studies along with their years. By the way, if you download the data, I've tracked down most of these papers. About a third of them were doctoral dissertations, and it's very difficult to find doctoral dissertations on the web from the late 60s and early 70s. So I didn't find them all. But if you download that zip file, it contains many of these papers, and you can look at the original raw information that this data set is based on, as well as the Rosenthal paper. And then we have the standardized mean difference and the standard error. So that's the raw data that we're going to be working with. So the first thing I need to do is meta set the data. So I'm going to use the declare generic pre-computed effect sizes option here. So I'm going to specify the stdmdiff variable as the effect size. That's the standardized mean difference. And then I'm going to specify the standard error for that mean difference. So if I were to, I can either use the control panel to specify that or type the command meta set stdmdiff and then se. So that's how we would set up the data. So if I type that command, it's going to give me the following output. It's going to summarize that I have 19 studies. The study label at the moment is generic, and it just kind of reiterates some of the information. At the moment, the model and method is going to be a random effects model using restricted maximum likelihood, again, because that's the default. Now, if I wanted to change that, I can come under the control panel here, right below where it says declare generic pre-computed. If I select the model button, if I click that, that will allow me to choose other options if I wanted to. So random effects or restricted maximum likelihood. I'm going to leave that alone because I'm just going to stick with the defaults, but this is where you would go about changing that if you wanted to. The other thing that I want to do, if I click the options button, this is still in that same setup page, I can click options, and that allows me to add a study label. So if I want to label the studies, it's kind of like adding a value label to a categorical variable. So for study label, I'm going to select the variable study LBL. And then for the effect size label, I'm just going to type std mean diff for standardized mean difference. So I can add those in here and click OK. So what that will do is actually update my meta settings. So meta update will update the meta-analysis settings and add a study label to it and add the effect size label as well. So all I've done is updated this. Whoops, I copied that slide in twice. So once I update this now, this information is a little more complete. It has the labels and things like that in here. So now I'm ready to start doing some summaries and actually doing some meta-analysis. So if I click on the summary button in the control panel, I can do some summaries. And this will allow me, if I want to change the meta-analysis model on the fly. I've already declared it as a random effects model, so I'm just going to leave declared model set. And so I don't have to type anything other than meta summarize. And what that will do is create a summary table of all of the studies, as well as an overall estimate of the effect size for all of the studies combined. But it gives me some other information, these test statistics at the bottom, and it gives me some heterogeneity measures at the top. So let's stop and take a look at this table in some detail. The effect sizes, what's shown in the main part of the table here, is for each study, it shows the effect size for that particular study, a confidence interval for that effect size for each study, as well as the percent weight. So the studies have different sizes. They had larger or smaller sample sizes, which leads to greater or smaller standard errors. And so Stata under the hood calculates the weights for you for each of the studies. How much weight does each study get? And we can see that some of these are fairly small. The Pellegrini and Hicks models have a weight of 1.69 and 1.72, whereas the Fielder et al. Study has a weight of 9.06, and the Evans and Rosenthal study has a weight of 9.06. So those studies, the Evans and Rosenthal and Fielder, are going to contribute more to the overall estimated effect size. So, and whereas the Pellegrini and Hicks studies are going to contribute less information to the overall estimated effect size. So at the bottom of the table, it says theta. Theta is the overall effect size that's computed from all of these effect sizes across all of the studies. So if we wanted to summarize all of these studies in one number, we would say that the standardized mean difference is 0.084. So that's the standardized mean difference, and we get a confidence interval for that right here. But the question is, remember we mentioned earlier, do we have apples and oranges? Are we simply making a fruit salad, or do we have really a bunch of good apples and we're making good apple juice here? So we need to pause and ask that question. Is it reasonable to combine all of these studies into a single effect size measure, that theta? And to answer that question, we can look at the statistics here at the bottom. So the first thing we really need to look at is this test of homogeneity, this Q value. So that Q statistic is a chi-square with 18 degrees of freedom in this case. So it's the number of studies minus 1. And that chi-square statistic equals 35.83. And we get a p-value for that Q statistic, and the p-value is 0.0074. Well, that's kind of bad news in our case, because that small p-value suggests that there's too much heterogeneity in our across these studies, and it's probably inappropriate to mix them all together. We've made a fruit salad rather than really good apple juice. There's just too much heterogeneity among these studies to be reasonable to compute them, to compute an overall effect size. So that means we really don't need to look at that point, we kind of need to stop and say, okay, this is not appropriate. We need to rethink what we're doing here. So at that point, we don't really need to look at this test of theta equals 0, because that's what that's testing. It's testing, is this summary theta effect size significantly different from 0? And we don't care, because we've just established that we probably shouldn't be doing that. Now, there are 3 other statistics that appear at the top of the table. So it's those 3 are tau2, or tau squared, here at the top, I squared, or it's listed as I2 in the output, and H2. So what are these statistics? Well, I've listed them. Oh, this is that test of homogeneity. This is the formula for it, if you're curious. And again, small p-values for that Q statistic suggest that it's not appropriate to report a common effect size. Now, the I squared, H squared, and S squared. The H squared, the tau squared, that's just the overall between study variance, just the raw between study variance. And it's really not very useful on its own, because it depends on the total variability. Just that number, it doesn't really have a context. So interpreting it is very difficult. I squared, though, is based on the ratio of tau squared, the between study variance, divided by the total variance, the between study and within study variance. So because we have these 19 studies, there's variability within the study that's captured by the standard error within each study, and there's between study variability. So I squared is the proportion of the total variability contributed by the between study variability. So I squared is that proportion. And if I squared is very large, let's scroll back up and look at that in our output. Here, I squared is 41.84%. What this suggests is that almost 42% of the variability in these studies is between the studies, not within the studies. So there's a fairly large amount of variability between studies. So that I squared is a kind of a descriptor of that. The Q statistic tests whether that's high, but I squared is a sort of way of quantifying that. 42% of the variability is between studies. H squared, I can tell you what it is. I just don't have a good intuitive interpretation for it. H squared is it's the sort of the inverse of that, or the reciprocal. It's the total variability divided by the within study variability. So if there's very little between study heterogeneity, then H squared equals 1. If there's a lot of between study heterogeneity, then H squared becomes greater than 1. But it doesn't have a nice interpretation as a percentage like I squared, which I think is why I squared is the more popular metric to report. But that's how H squared is defined. It's the total variability divided by the within study variability. And again, it equals 1 if there's no heterogeneity. So Q is the test, and then these are the descriptors for we're exploring the between study heterogeneity. There are also measures for fixed effects models, and they're related to these, but we're not going to go through all of the math. So we've established that we might not ought to be reporting an overall effect size. But just so we're for completeness, let's take a look at something called a forest plot. If we click on the forest plot tab, what it will do is show us essentially the same information from the table in a graphical format. And often it's a little easier to see the heterogeneity in these forest plots. So what this is, for each of the studies, it's going to show a box. a box with a little green line. So the box will be centered at that. If you look at the very bottom here, it shows you the effect size scale, 0 in the middle, left is negative, to the right is positive. The size of that square, the location of the square is where the effect size is, left or right, and then the size of the square is proportional to the weight. So notice that the Rosenthal study here at the top has a fairly large square, and if we look at the weight column, it has a weight of 7.74. Those Pellegrini and Hicks studies with a fairly small weight, they have a fairly small square because they have a lower weight. Now, also notice that the studies with the smaller squares also tend to have wider confidence intervals, and that's because they have smaller sample sizes and larger standard errors. So it's typical to see studies with smaller squares having wider confidence intervals. Now, what's interesting about forest plots is this makes it a little easier to just scan down this column and see that the effect sizes are fairly heterogeneous. Some of them are very positive, and some of them are a little bit more negative, and some are sort of in the middle. So there is this heterogeneity, and it's a little easier to see that visually using a forest plot. And again, the statistics are reported here at the bottom. We have the heterogeneity statistics, the tau squared, I squared, and H squared, and we have our Q statistic here at the bottom. So this is very much like the table that we saw earlier with the addition of this graph. But again, the graph makes it a little bit easier to visually see the heterogeneity among the studies. So what are we going to do about the heterogeneity? So Raudenbush suspected that the amount of time that the teachers spent with the students prior to the experiment might actually influence the results. And so one of the things they wanted to do was consider this. So how are we going to do this? One way we can explore the heterogeneity, if we have a categorical variable that we're interested in, is to do a forest plot using a subgroup. So in this case, we can check the little box that says subgroup meta-analysis and then specify a categorical variable like week 1. Remember, week 1 is student-teacher contact greater than 1 week. So what we're going to do is recreate that forest plot in two groups. So I'm just going to click over here so we can see the forest plot itself. The top part of this forest plot is for studies where student-teacher contact was less than or equal to 1 week. And then the bottom part of the forest plot here is for student-teacher contact greater than 1 week. Well, notice in the bottom set, those boxes tend to cluster together a little bit better. There's not as much left-right heterogeneity among the results. Whereas the studies where the student-teacher contact was less than 1 week, those tend to be more heterogeneous. And in fact, we get a Q statistic for each of these groups. And so in the top, we see the heterogeneity, the I squared is 22%. So 22% of the total variability is between groups. And that Q statistic now has a p-value of 0.13. So now it's greater than 0.5. Wait, let me make sure. Yes, that's the Q statistic. So that Q statistic, it does have a p-value greater than 0.05. If I look at the bottom group for the teacher contact greater than 1 week, that I squared is 0. So it's suggesting that almost none of the variability is between groups. And if I look at the Q statistic below that, that Q statistic p-value is 0.78. So if teacher-student contact is greater than 1 week, the heterogeneity drops off a lot, which means it's probably more reasonable to report an overall effect size for the studies that reported greater than 1 week, teacher-student contact greater than 1 week. At the bottom, it reports the overall tau squared and H squared and Q statistics. So we get all of that reported in one forest plot. Okay. Now, what if we wanted to treat that teacher-student contact as continuous? So we can explore heterogeneity with continuous variables. They call them moderators in meta-analysis jargon. So they're referred to as moderators. I could do this using meta-regression. So if I click on the regression box in the control panel, this allows me to specify a moderator or a study level covariate that may influence the results. The command here is meta-regress. Stata already knows, because of meta set, it already knows what the outcome is. So all we have to do is type meta-regress and tell it the name of the moderator or the variable that may influence the study results. This is then going to, the command is meta regress weeks. And this looks very much like regression style output. It shows an intercept, but more importantly, it shows a coefficient for weeks. And what this suggests is that for each additional week of student-teacher contact, that effect size is decreasing. There's a downward slope. And we also get a report of the tau squared and I squared and H squared and so forth here, as well as that residual test of homogeneity. So what does this mean? Well, what we can do is create a bubble plot. So after we fit our regression model, you can click on the post estimation button, and this opens the post estimation selector, and we're going to create what's called a bubble plot. And the command is just estat bubble plot. And what it's going to do is it's called a bubble plot. So here the horizontal axis is that moderating variable, the weeks, the actual number of weeks of prior student-teacher contact, ranging from 0 to 25 weeks. And then the vertical axis is the standardized mean difference. That's that effect size that we're studying here. And we can see the green circles here are the individual studies. They're the individual observations. And it's called a bubble plot because the circles are weighted. The size of the circle is proportional to the weight of that particular study. So smaller circles indicate smaller weight studies and larger circles indicate larger weight studies. And we can see that the line has a downward slope to it. And this suggests that more weeks of prior student-teacher contact is associated with lower effect sizes, okay, that relationship. So this suggests that there is some effect of teacher-student contact on the results. So there's that. We can't just ignore that if we're going to talk about a summary statistic. So we'll come back to that here in a bit. Another thing that we probably need to explore is publication bias. It's entirely possible one of the reasons for our different results could also be because we're all probably aware of this, that only studies that are usually studies that have small p-values are published, and studies that have larger p-values, typically greater than 0.05, are just not published. And so when we do a meta-analysis of published studies, it's very likely that we're only going to see studies that have p-values less than 0.05, that show some sort of an effect. And we're never going to see the studies that showed no effect because they were never published. And so this is called publication bias, and we want to explore that. A way of exploring this publication bias is to create something called a funnel plot. And I'm not going to read this entire slide to you, but it's easier to show it in an actual picture. But what we're going to do is click on the publication bias button here on the control panel. And for now, I'm just going to create a standard funnel plot. So I'm going to select that. That will issue the command meta funnel plot, and it's going to create this thing called a funnel plot. So the horizontal axis here is the standardized mean difference. That's our test statistic. The vertical axis is the standard error. Oh, excuse me. Sorry for the coughing. The vertical axis is the standard error. Now, keep in mind the standard error is it is mathematically proportional to the sample size. And it's in reverse. That vertical axis is reverse scaled. So notice the top is 0 and the bottom is 0.4. So the green dots then are the actual studies themselves, and they're plotted at the point of the standardized mean difference and their standard error. So studies closer to the bottom have larger standard errors, which probably translates into smaller sample sizes. And notice that we typically see where the studies that have smaller sample sizes, it's not unusual to see those studies only on the positive side, show studies that show a positive effect. Now, why is this important? Well, studies that have a small sample size probably lack the statistical power to detect small effect sizes. And so if we look at this plot, most of the plots fall inside of this sort of pseudo 90%, 95% confidence interval. But we do see a couple of these studies over off to the right that are fairly small studies. They had large standard errors, but they had very large standardized mean differences relative to the other studies in the sample that we collected. And notice that we don't see those on the negative side. So these are some outliers. These two studies here, and we could identify which studies those are, those two studies are kind of unusual. And the fact that we see those on the right side and we're missing things in this area to the bottom left suggests that there could be some publication bias. Publications that are not showing an effect, that have p-values less than 0.05, are not being published. We're not seeing them. And studies that do have these large effect sizes had large standard errors. So those may be just flukes in the literature. There's a way to actually test for this. It's called, one of the tests is called an Egger test. It's a regression-based Egger test. And so if I select that test, or I could type the command meta bias Egger, it will actually test and put a p-value on whether or not it's likely that we have this small study effect. And sure enough, if we look at the results at the bottom of the test, the null hypothesis is that beta 1 equals 0, that there's no slope here, there's no small study effect. And it reports a p-value, and in this case, the p-value is 0.01. This suggests that there is a small study effect. And visually, we can see that here in the funnel plot, but the test then kind of confirms that with a test and gives us a p-value that we can report for that, okay? Now, there are a variety of these tests, and Egger is the default, but there are some others that, again, are used in certain circumstances, and I'm not going to go through all of the options and why here, but it's all in the manual if you want to read more about it. So you have other options besides Egger if you want to use them. You can also add contours to your funnel plot, and you can specify the significance contours. So you may want 99% confidence intervals, 95%, and 90%. So you can add contours to those funnel plots. So here on the outside, we can see this gray area is the 1 to 5%, and then we have the 5% to 10%, and then the greater than 10%. But again, the results suggest the same thing, that we have this issue with small study or publication bias. Now, again, we can check for this accounting for weeks. So I could create this funnel plot separately for whether the teacher-student contact was less than 1 week or greater than 1 week. So here I'm going to create those funnel plots separately. And now when I take this into account, notice that the studies where the student-teacher contact was greater than 1 week, those studies all cluster nicely right around the overall effect size, and they all stay within that 95% confidence. I call it a cone, but it's an interval of sorts. The ones where we have the more variability are in the studies where teacher-student contact was less than or equal to 1 week. So that seems to be where we're having more of this issue. These small studies with small sample size and have teacher-student contact less than or equal to 1 week, those seem to be driving this publication bias issue. And then if we wanted to, we could go through and do this Egger test accounting for week. So I could type meta bias and then tell it week 1. This I.tell stated that that's a categorical variable, and I want to report the Egger test. And now when I account for study for the teacher-student time, that week 1 variable, notice that that goes away. So accounting for the student-teacher contact, that seems to be what's driving this small study effect or publication bias. So it's, again, another way of identifying what's leading to the heterogeneity in these issues among the data. So if you do suspect publication bias, you do want to take a look at these other factors and see it may be heterogeneity, not publication bias, that's driving these patterns in your data, so in your results. Now, there's another thing called meta trim fill, and you can it's a kind of like imputing the data. And I'm not going to spend a lot of time on this, but just be aware that you can do this. It's called trim fill, and it is a kind of imputation. And essentially what it's doing is filling in what would those studies have looked like. What would the studies have looked like had they been published? And so essentially, if you stare at this long enough, these 3 points in red on the left are sort of the mirror image of the most extreme points on the right. So the ones on the left in red are the imputed studies, and that's what we expect that we might have seen had those negative studies actually been published, and we would have seen them in the literature. Okay. Now, this is something that I kind of think I don't know why we don't just start with this. It's called cumulative meta-analysis, and cumulative meta-analysis performs the meta-analysis by accumulating the studies one at a time, ordering them with respect to one of these moderating variables or study level variables. So what you can do is go back to forest plot, and there's a little checkbox here that says cumulative meta-analysis. And then you can tell it the ordering variable. In this case, I want to order the studies by the weeks of student-teacher contact, and I want to sort them in order ascending. And so the command then would be for meta-forest plot cumulative weeks. Now watch what this does. If I look at the far right-hand column in the table here on the far right side, it's showing the number of weeks of teacher-student contact. So when the weeks are 0, notice it's ascending. So it's 0, 0, 0, 0, 0, and then 1, 1, 1, then 2, 2, 2. And then by the bottom of the table, we're getting to 19 weeks of student-teacher contact, 21 weeks, 24 weeks. And look at the results in the forest plot. The pattern just jumps right out at you visually that the more weeks of student contact, the lower the overall effect size. I mean, the pattern just is obvious when you do this cumulative forest plot. The pattern, to me at least, is obvious that the weeks of contact have a direct effect on the overall effect size. And the more teacher-student contact, the lower or closer to the null the overall results. Okay. So clearly there's this heterogeneity, and clearly the pattern is more weeks of student-teacher contact decreases or drives the test statistic towards the null for each of these studies. Okay. So that's probably what we would end up reporting here, is that there really isn't a big effect. If we were going to answer the question, is there a relationship between student-teacher contact and IQ test? The answer is probably no. If we gather enough data and allow for enough student-teacher contact, there really isn't much of a difference. The differences seem to appear in these studies with smaller sample sizes and less actual weeks of student-teacher contact. So it's less about the effect and more about the amount of time, the contact time. Okay. Now let's backtrack. I mentioned earlier that we had 3 options for looking at meta-analysis, the meta-analysis models, and there are 3 of these. The first one is called the common effect model, and notice it's singular, common effect. And so here the overall theta, the overall estimated effect for across all of the studies is assumed. What we're going to assume is that there really is a true effect, and that each of the studies just deviates from that true effect. So there's an error of each of these studies deviating from the one true effect overall. So that's the common effect model, and it's a little bit, it may be true in certain circumstances, but it seems a little unlikely that there's really a true effect, a common effect. The fixed effects model is a little bit different. Essentially what it's saying is that our overall estimate of theta across all of the studies is equal to, each study has its own effect. There is a unique effect for each study, and that each study has its own unique error. And when we average across all of those, we get this fixed effects model. Now the random effects model, what we're assuming is that overall effect, and I think this is why it's so commonly used, is because it's a little more realistic, that our overall measure of the true effect, theta j hat, is equal to some true theta, that there really is a true theta in the population. But there's a deviation, a uj deviation for each of these studies from that true effect, and that, or between study effect, and then there's an error of each study from that true effect. So there are 2 sources of variability. There's the EJ is the within study variability, and UJ is the between study variability. And but there is some true overall effect that we're trying to get at. And I think that's why the random effects model is so common, because I think it describes probably more accurately what we really believe about the effects in these studies, that there is some true effect, but that there's going to be variability both within and between studies. So that's why it's so commonly used. I put these in, I think we're running out of time, and I don't want to dwell on the math too much, but they're common estimators, and you really do get pretty similar results, even though the mathematical assumptions are a little bit different. So I think I'm going to wrap up here. Again, the meta-analysis suite that became available in Stata 16 allows you to conduct your entire meta-analysis within Stata. And again, we've used meta set to make it a little easier to type the commands. So you describe the structure of your data once, and then you just use the other commands to explore the heterogeneity and things like that. So if you'd like some additional resources, there's a quick overview on the Stata website of these. There's a full list of meta-analysis features also on the Stata website, and these are hot links within the slides. So if you download the slides, you can just click on them. There's a YouTube video on meta-analysis up on our YouTube channel. I based the original version of this talk very heavily on Yulia Marchenko's original talk from the 2019 UK user group meeting. So if you'd like to see another version of this talk, there's one that she did a few years back. And mine is very similar, but with some additions to it. Again, if you'd like to actually look at more actual meta-analysis studies, go check out the Cochrane Project. It's an interesting website, and you can search all kinds of different topics and things like that. And I've also included a list of references here at the end in case you want to look them up. So thanks for joining us today. I hope this was helpful. And again, if you want to download the slides, data sets, and do files, that's all available at tinyurl.com/Stata Meta-Analysis. And I'm happy to hang around and answer questions for a while. And if you think of them later on, just shoot me an email at chuber@stata.com. So thanks again for having me, and I'll jump off and answer questions.
Thanks, Chuck. I'm going to stop recording so that we can have a smaller video. So here we go. Or pause recording, maybe.
Okay.